Alexander, who likes to be called Sasha, is co-director of informatics at the Harvard Personal Genome Project, abbreviated PGP. Some of you have heard that name throughout this conference. He is Chief Innovation Officer at the Curie Corporation and founder of the Arvados Project. The Arvados Project is a free and open source software platform that facilitates development of clinical quality applications for managing and processing massive data sets, spanning millions of individuals across collaborating organizations and eventually encompassing exabytes of data. So his talk title today is Expanding Sequencing, Phenotyping, Interpretation, and Biobanking of the Personal Genome Project. Go ahead and take it away, Sasha.
Thank you so much, Eric. Can you hear me okay?
Yes, we can hear you.
That's great. So I'm going to tell you a little bit about what the PGP is and how we're currently expanding the project. And first, I'm just going to motivate us a little bit. So what we want to do is we want to make precision medicine possible for everyone. We all know that genome sequencing has been dropping exponentially in cost, especially human genome sequencing. But how are we going to get genomes and genomic medicine to millions and eventually billions of people? We have algorithmic challenges, there's compute and storage challenges, there's all sorts of regulatory and privacy concerns. Patients also want to have more say in how their data are used, and there's a huge lack of standardization. And this has been a huge problem for many decades, and of course, when genomes were millions of dollars, people thought this was kind of an esoteric problem, but it's not esoteric anymore. And when I think about this, I like to borrow from free software. So free software has existed now for many decades. It powers Linux, it powers Android, powers alternatives to Microsoft Windows. Free software has expanded to include free knowledge, which includes projects like Wikipedia. And so could we use ideas and the sort of free software ideals to bring precision medicine to everyone? So to do this, and it is actually a little bit complicated, software, integrated human data set, biomaterials, other information, each of these things need their own legal constructs to make them open and to make it possible for other people to reuse work. And if you just ask your general counsel to go and do this for you, they will come up with some reasonable legal construct, but is completely incompatible with everybody else's legal construct, and you'll be stuck. And so we've spent a lot of time thinking about how do we do this in a way that lots and lots of organizations could build an ecosystem that's open. And we've implemented all of this at the Harvard Personal Genome Project. George Church started this in 2005 at Harvard. The project is now global. I started with the project pretty much at the very beginning, started recruiting folks. I was one of the first people to get the general public license, which is a common open source license, approved at the Harvard Office of General Counsel. And that allowed the project to start sharing our software in a way that lots of other people could use. The PGP is freely available. It brings together genomic, environmental, trait data, other biomedical data, all from openly consented volunteers. Currently, there's about 500 whole genomes at the Harvard PGP. Other international PGPs have more whole genomes. And there's about 100 cell lines at Coriell and Neurostem Cell Foundation. And these are all used by open standard efforts such as Genome in a Bottle, which notably has the Ashkenazi trio and the Chinese trio, all from the PGP. So currently, our expansion efforts are focused on making better interpretations of these data that we can share with participants, increasing the diversity of both sequencing and biobank samples, and also making our phenotyping better. We've been doing a study since March focused on COVID-19 and phenotypes related to that. And so that's kind of where the PGP is right now. In 2012, when we first published the 10 participants, the first 10 participants of the PGP, we focused on sort of a very simplistic interpretation. You know, you look at a Mendelian gene, you see a well understood by clinical labs Mendelian variant, and you say, hey, you know, you have this thing. And we actually had a reporter who was very public, as a Pulitzer Prize winning reporter, John Lauerman, who actually had a quite concerning variant that came up in his workup from the Personal Genome Project. So by doing that, you know, we sort of pioneered the full cycle of consenting participants, phenotyping them, sequencing them, and returning data. But what if we really want to use the whole genome? What if we want to use the portions of the genome that the Telomere to Telomere project is starting to generate, that regions of the genome that we've really never seen before? Well, how would we take advantage of those regions? So our thinking about that is to look at machine learning. And we've been doing machine learning now for many, many years. It's a great way to form relationships between genome and phenotype. We can look at things like visible characteristics such as eye color or blood type. We can look at current health conditions, all sorts of things. And then you can use these machine learning predictors to make, to do drug discovery or target identification for drugs, look for risk factors, all sorts of things, all of the usual things. But in the PGP, all of the data is completely public. You don't have to talk to us, you can just download it right now. So why do this in the PGP? It's a great source of openly consented genetic and phenotypic data. We can focus on strongly penetrant traits such as eye color to make sort of baseline ML models, and then we can start working on more difficult traits as we get good at the easy stuff. So here's a predictor that we've made for blue, not blue eye color. This is just with 100 genomes labeled with blue eyes, not blue eyes. This predictor with just 100 genomes and fairly standard machine learning techniques immediately pinpoints two well-known in the literature important variants. So for us, we were really amazed and excited by this because there's only 100 genomes. And so it's quite repeatable. You can do this with blood type, you can do this with all sorts of things. The model, don't have to go into details, but the reason that these models are interesting is that they're so generalizable. We use a representation called tiling, where we normalize all of the genetic data, which allows us to use data that's exomes and microarrays and all sorts of data from different sequencing technologies. Then we one-hot encode the genomes, we filter them, and we use a GLM with regularization, and that gives you the eye color predictor I just showed you. And again, what's great about this is that you can apply it to lots and lots of phenotypes. And you can use it in closed cohorts where it may not be so easy to do method development. So by itself, an eye color predictor isn't very interesting. But because the PGP participants are recontextable, we can lean on our open consent methodology, which was developed by Jean-Tine Lonchoff and others in 2007, and then approved by UK and Austria and Canada to go back to participants and say, hey, we made this predictor. Why don't you give us some more phenotypes so we can make more predictors? And where diversity really sort of fits into this is that if you make a predictor in a cohort that's predominantly one ethnicity, and then it turns out that predictor doesn't work at all in some other cohort with a slightly different ethnic mix, then that's not very good. That's not going to be precision medicine for all. And so in a way, this gives us a tool and potentially gives the Pangenome Consortium a tool to think about, how do we know that we're doing a good job of including lots of diverse genomes in our pangenome, because we want to be able to create these predictors that work, for everybody. And maybe there are some ethnicities that don't want to participate in the pangenome, but these predictors can still work for them. And so that's something we're going to have to figure out. When does it work? When does it not work? How many genomes do you need before these methods don't work anymore? There's all sorts of details that you just couldn't figure out unless you had data to play with. So in the PGP as it is today, there are trios, there are participants with genotyping and whole genome sequencing, and there are participants who have given us self-identified ethnicity, roughly 500 individuals, all of whom seem to have a reasonable diverse, sort of non-white cohort that we could go talk to and sequence and add to the biobank. So these folks have less than 50% European ancestry, or using genetic data, or using their self-reported ancestry. We found self-reported ancestry works very well for matching what you would predict from genetics, except for folks that are white plus American Indian and Alaska Native. And there, I think there's all sorts of complex issues that make this a little bit more challenging, or actually much more challenging. And so we're actually very excited to learn from the consortium about the sort of learnings from communities that would potentially never participate in the PGP and would have to be in more closed cohorts. All of these analyses are available in Jupyter notebooks, and so we can work with the consortium or really anyone that wants to improve the diversity of the Personal Genome Project and how we think about diversity in the Personal Genome Project. So this is just a plot of participants in terms of our computed diversity. And again, these would be folks that we could prioritize for sequencing and biobanking and phenotyping. So our current plans are to collect saliva from all participants with low coverage, and then do sequencing with low coverage, long read sequencing, and then do optional deep sequence, short read sequencing as sort of resources permit. BGI has actually offered to sequence up to 10,000 participants if we send them DNA. And so we're excited to take up that offer. The plan is to collect blood by mobile phlebotomy from prioritized participants, then just make LCL lines that are kind of familiar to the consortium that are all at Coriell. And currently we're doing this as a pilot in a small COVID-19 project. And that's a summary of where we are. So we'd love to get some questions. You know, we're very excited about learning from the consortium.
Well, thank you, Sasha, for your great talk here. You covered a lot of different areas. I'll go ahead and ask the first question, and wondering, so taking off that comment where you said you didn't select individuals that were admixed between Native American and European. And what was the reason for that? And was it amount of admixture, or was it just confusing to begin with?
So the issue is that if the people who said they were white and Native American, but also provided us genetic data, it was indetectable.
Yeah.
And so I think that's a well-known thing, because we don't consider that genetics are not important for whether or not you're Native American. Yeah. So how to deal with that is just.
Let me quickly add to that. Do you think some of the issue could be that there's not enough sampling of Native Americans to have SNPs that you can recognize in a database, or you think it's a misperception of ancestry?
Well, so I think my opinion doesn't really matter. I think this what we have to work with Native American communities to answer that question in a reasonable way. Because I think it's really offensive for people to come into this and be like, well, I'm going to speculate about whether or not you're Native. Like, that's just, you know, we shouldn't do that.
Right.
So I think we have to work with the communities, and if they want us to do that, because I think that's plausible, that actually we would detect it if more people participated, then yeah, that would be fine to do. But we shouldn't do that unless the community wants us to do it.
Okay. All right. Yeah. Good. Thanks for answering that. So we have some questions here coming through. Zhao Yu Zhu says, why use low coverage sequence reads? And I'll add to that, you know, what the BGI volunteered data set and others, are these short reads or long reads? And is this a sampling, or is this actually to get a de novo assembly?
Right. So if people have seen, there was very impressive low coverage polygenic risk score work from Ehrlich's group at one of the ancestry companies. Basically, you can take low coverage genomes and very inexpensively start to make polygenic risk scores. So our thought on this topic was that eventually we're going to have cheaper technologies that give us end-to-end genomes, which you guys are actually making here. Why don't we do low coverage, long reads inexpensively, which will give us some interesting insights into structural variants, methylation patterns, some other things. And then that will give us enough information to prioritize people for deep sequencing. Because in my experience, we've now, you know, we've done deep sequencing for many, many years. And it's roughly 10% of people have something that you can say about their genome. So if you have nothing much to say, could you get at that answer more cheaply and only prioritize people that may have a concerning finding, or you may have something to say?
Okay, for the sake of time, let me continue on with the questions. I know I asked a few there myself, but let's give the audience a chance, and we have only 2 minutes left. So how do you square this from Dr. Novak? How do you square the benefits of open licenses for data from people who feel society already protects their interests with the need for close community supervision of the data, use that is, from communities that have good reason to be suspicious of would-be data users.
So our feeling on this is that the PGP is international because there are privileged people of every ancestry. And so one of the ways to find those people is to just go to their countries and have PGPs in those countries. And the PGP is going to have lots of gaps. You know, we would like to make all of our methods as open as possible and to just show those gaps as in as glaring and as honest a way as we can. So we don't feel that you have to automatically enroll underprivileged people in the PGP. Like, it's just not a good idea. But if you have an informed consent process and you find people that feel that they are appropriately privileged to participate in the project, and they're comfortable with the risks, then, you know, we should find people of every ancestry that we can.
Okay, quick one, last one, and then Ramal, I will take your question to the panel later, but also from Novak. Isn't predicting non-medically relevant but socially important phenotypes like eye color potentially dangerous?
What we feel at the PGP is that it is insanely dangerous that a bunch of companies have 30 million people in their genetic databases, and the vast majority of the public, and that includes entire countries, are more or less locked out of that activity. And so we think it's more important, given how pervasive genomic medicine could become, that there be a public resource that people can draw on.